Hello everyone, today we will discuss about factors that affect enzyme activity. There are many factors that can influence enzyme action in any chemical reaction. First of all, we will discuss about temperature. Like any chemical reaction, the rate of enzyme controlled reaction also increases when we increase temperature. So, with the when molecules they're moving fast, for example, this is substrate and this is enzyme, so the chances of their collision to each other they increase. So now they are colliding with each other. It is more likely that substrate molecule this get into the enzyme active site to make an enzyme substrate complex. So what is actually happening with the increase in temperature, we are increasing the chances of making ES complex. But if temperature gets too high, the reaction stops. So if we see a graph between temperature and rate of reaction, we see that with increase in temperature, the rate of reaction it increases. With every 10 degrees centigrade increase in temperature, the rate of reaction it doubles. For example, this is point A and this is point B. So from point A to B, so the substrate and enzyme molecules they are binding to each other to make ES complexes, enzyme substrate complexes. So here comes a point where the rate of making these complexes is highest. So we call this temperature as optimum temperature. Beyond this point, if you keep on increasing temperature, now reaction won't increase, the reaction rate starts to fall down. And there comes a point where reaction stops. So now what is happening here? So increase in temperature now, breaking the bonds that hold the enzyme in shape. So increase in temperature is now destroying the structure of enzyme. When structure of enzyme is destroyed, so its active sites are not available for substrates. So substrate can't bind to active sites to make an ES complex. So enzyme is not able to perform its activity. It is not able to catalyze the reaction now. So at this point we say that enzyme is denatured. So next factor we'll discuss about pH. Let's see if pH affects the rate of reaction. So all the enzymes they work at their optimum pH. So this is the optimum pH. So below or above this optimum pH, the enzyme is denatured. Means it is not available to perform its function. It won't be catalyzing the reaction below or above its optimum pH. So what is actually happening here? If we do below, it means we are increasing the hydrogen ions as in acids, and if we do above, it means we are increasing the hydroxyl ions as in alkali. So these ions, hydrogen and hydroxyl ions, they actually mess up with the hydrogen bonds and ionic bonds that are present in the structure of enzyme. So now these ions they will destroy active sites. So substrate won't be able to bind to the enzyme active site and won't be able to make an ES complex, and reaction won't be catalyzed. Most of the human enzymes they work at pH 7. This is the neutral pH. There are some exceptions, for example, pepsin. It works best at pH 2, that is acidic. There is another example, trypsin. This works best at pH 8. This is slightly alkaline. So next we will see about enzyme concentration. Does it affect the rate of chemical reaction, enzyme catalyzed reaction. Yes, when we increase enzyme concentration in a reaction, the rate of reaction increases. So now what is actually happening, when we add more enzyme, it means that we there are more active sites available for the substrate that they can bind to. So with the increase of enzyme, actually the rate of reaction it increases. But if the substrate is limited, then there comes a point, for example, from point A to B. Here, more substrate, more enzyme you will add, there are more substrate molecules that are available in the solution. It will bind to the enzyme and the rate of reaction it goes up. So but when all the substrate is used up, now at this point, if you add more enzyme, there are more active sites available, but there is no substrate. So adding more enzyme here from point B to further C, it doesn't make any difference. So next factor is substrate concentration. So if we increase substrate concentration, the rate of reaction it is increased, because now more substrate molecules if they are available, so it is more chance to for substrate molecules to collide with each other and to make an ES complex. But there comes up, for example, from point A to point B. Here the more substrate is binding to substrate available to bind into enzyme active site that are available. So now at point B, what is happened here? All the active sites on the enzyme are full now. So there is limited enzyme now. So we don't have any more enzyme. And at this point, if you keep on adding substrate, so substrate won't be find any free active site to get attached with. So adding more substrate at this point from point B to C, that doesn't make any difference. So with the passage of time, rate of reaction it is observed that it is decreased. Why? Because the substrate molecules they are being used up and they are converted into the products. So in this case we can say the starting point, the starting rate of reaction or the initial rate of reaction is the highest rate of reaction. So enzyme activity can be inhibited by adding certain substances. We call them as inhibitors. They stop the activity of enzyme. So there are two types of inhibitors: competitive inhibitors and non-competitive inhibitors. So first we'll talk about competitive inhibitors. These inhibitors they actually compete with substrate molecule to get into the active sites of enzymes. For example, this is enzyme and this is its active site. This is inhibitor and this is substrate. So these, because as the name indicates, competitive inhibitors, these actually they compete with each other, substrate and inhibitors, to get into the active site. So the inhibitor, when it is attached to the active site, it is actually blocking the active site. So substrate molecule they can't get there and they cannot make ES complex. So these inhibitors they are quite similar in shape with the substrate molecules, and that's why they just fit into the active sites and block the activity of enzyme. How much activity of enzyme is inhibited, it depends upon the concentration of the inhibitor and the concentration of the substrate. If the concentration of inhibitor is more than the concentration of substrate, so now there are more chances of inhibitor to get to the active sites. But if the concentration of substrate is more than the concentration of inhibitor, then the chances of substrate to get into the active sites are higher as compared to the inhibitor. Graph shows a link between substrate concentration and rate of reaction. So this is the rate of reaction without an inhibitor, and this is this line is showing the rate of reaction with inhibitor. But if we add more substrate here, so rate of reaction it then further goes up with the increase in substrate concentration, because now the chances of substrate molecule, because of its higher concentration, are higher to get into the active sites. So next we will discuss about non-competitive inhibitors. So non-competitive inhibitor, as the name indicates, they don't compete with the substrate molecule to get into the active sites because they are different shape. For example, this is the enzyme molecule and this is the substrate molecule. So the inhibitors, because they are different shape, so they get attached to the enzyme other than the active site anywhere. All the other site other than the active site, we call them as allostatic sites. So these are attached here. So when these inhibitors, they bind on the enzyme other than the active site, what they do, they just change the structure of active site. So when active site is changed because of the attachment of inhibitor, now substrate molecule won't be able to attach to active site and no ES complex can be formed. And when no ES complex can be formed, so enzyme can't catalyze its reaction. So this inhibitor is blocking its activity this way. Now, if we see a graph between the concentration, like this graph is showing if we increase the concentration of substrate, does it make any difference? No, because when you increase the concentration of substrate, it has no effect because the active site is no available. Adding more substrate will make no change in the reaction. So reaction won't be catalyzed.